Hello everybody. This is strange, isn't it? I'm very sorry not to be able to join you all in Finland, but I just want to say thank you for tuning in for this talk on cortical depth across the brain surface. So why are we doing this? Well, I guess we're all here at this meeting because we believe in some way that brain stimulation, whether we're talking about TMS or one of the electrical modalities, has some role to play in helping people to do things better. So whether we're talking about fixing neurological problems, or maybe we're talking about cognitive enhancement in otherwise healthy people. But we have some challenges, and I guess the biggest set of challenges really are about targeting and setting dosage for brain stimulation. And there are lots of different ways of thinking about that, but effectively we're talking about the ability to decide where and when to deliver our stimulation and how much of it we should give. One problem in dose setting is that we don't really have a good model of how electrical or magnetic energy affects the brain, or at least gets from the stimulator to brain tissue and thereby has an effect. Now, we know, I guess, the physics behind all of this, so we know there are things like Maxwell equations and Lorentz laws and things. And when we know some of these principles, we can combine them into computational modeling techniques. So you'll see many beautiful images like these finite element models that are produced. I'm sorry for not crediting this. This is from Andy Huang's ROAST toolbox for MATLAB. One thing that we that adds variance to our models is cortical depth. So why does that matter? Well, we know that at least for TMS experiments, the amount of extra output you need from the stimulator to have the same effect, given a difference in cortical depth, is about 3% per millimeter. So if the brain area that you're talking about or trying to target is 1 millimeter deeper than some reference level, then you need to turn your dial up by an extra 3%. So there's some clever work over the last few years by Mark Stokes and colleagues that helped us to understand the role of depth itself, so irrespective of the intervening tissues. My interest here is actually in the variance of cortical depth. And in fact, not just variance, but the coefficient of variation. So the coefficient of variation is the standard deviation divided by the mean, and it's scale free. So for most distributions, the standard deviation or the variance should increase as the mean increases. That's not true for for coefficient of variation. So that allows us to take out the effect of different depths of cortex and to replace that with a scale-free measure. And why would we do this? It's because if we find a brain area that has relatively low variance or low coefficient of variation, we can use reasonably simple methods for targeting that area or for dose setting for that area. Whereas if a brain area has relatively high coefficient of variance, so if between participants it's if it's shallow in some people and deep in other people, then we have a problem and we need to use more specific methods to target that particular brain area. Okay, how have we done this? So this uses some pre-existing data. In fact, these were taken from the OASIS database, which is freely available. There are something over 400 images in the OASIS 1 series. I was only interested in people in their sort of middle years, so 25 to 60 years old, where the structure of the brain is relatively stable, and people with a dementia score of 0. The full methodology is described by Marcus and colleagues in a paper that's cited here. But we have 94 images in this database, so that's the number of images that contribute to what I'm about to show you. Okay, image processing. Right, there are a couple of steps here. So firstly, SPM12B was used to segment the images into the two parts of the image that are of most interest, and those are the grey matter, so the place where we assume TMS and TDCS are having their effect, but also the soft tissue. So the soft tissue layer in SPM segmentation includes the scalp. To find the scalp surface itself, we can just apply a 3-dimensional convex hull, so it just fits the minimum frame that fits the outside of the soft tissue layer. So that's a proxy for the scalp layer. And then from there, for each point in the grey matter image, or the grey matter segment, we can find the minimum Euclidean distance from that point to a point on the scalp surface. So where is the closest point on the scalp to that brain region? So we do that for each person. We can then take those distance images and transform them into a standardized space, in this case, MNI space. And having everything in a standardized space means that we can do group statistics on everybody's image all in one go. So we can, in this presentation, I'm only going to talk about the coefficient of variation and some correlations. Okay, so what do we find? Well, one thing we would look for in our group data is whether the group data matches what we expect a cortex to look like. This is an inflated image of the brain. So we've taken the mean of all of the MNI format images and just put them all together and found the mean depth of each of those voxels. Fortunately, it looks roughly the way we would expect. So a gyrus, which is relatively close to the brain surface, to the scalp surface, should be in a relatively bluish color, so a smaller number, whereas deeper areas, particularly folded underneath the brain, under the temporal lobe, we would expect those to be deeper and a redder color. So that's reassuring. Just to orient people who aren't used to looking at inflated brains, the central sulcus is indicated here, and it should be relatively deep compared to the precentral and the postcentral gyrus either side. Now, if we talk about the standard deviation of depth, now the pattern isn't quite so obviously mapped onto cortical folding. Why would that be? It means that there's some variance introduced into this measure that is not related primarily to depth. And the way to spot that is to divide the standard deviation image shown here by the mean image shown in the previous slide to get this image, which is the coefficient of variation. Now, the coefficient of variation suddenly has a much wider distribution across this brain surface. And this reflects two things. So firstly, there's a process variance, so the variance in warping each individual brain image into a standardized space. So SBM works very hard and is very good, but there's going to be some variance introduced there. But also there's some source variance, so variance in the folding of different people's cortex. And I think between these two images, there are ways to tease these apart, but these are the sources of variance that we would need to worry about in talking about standardized brain spaces. Okay. Now, just to make things a little bit simpler, because that's a complex image, we can look at regions of interest. And so if we take some commonly stimulated regions of interest, so there's a middle frontal gyrus here, there's a precentral gyrus, angular gyrus, the superior occipital cortex gyrus, and superior temporal gyrus. These are brain areas that contain interesting functional areas, and so are commonly used in brain stimulation experiments. So looking at the coefficient of variation of these regions, well, there's some variance across those areas. So, for example, the occipital cortex has a relatively high coefficient of variance, and also seems to be different between the left and right hemispheres, which however relatively low variance in the prefrontal cortex, and that's a bit more optimistic for example, cognitive experiments where we want to knock out or enhance some kind of cognitive processing. Or this is a common target for depression-related targeting as well. Now, thinking about correlations in depth, well, one thing that will be a good sanity check is whether brain or head size correlates with age. The size of the head does not correlate with age, so your head does not shrink or grow as you get older. But your brain volume does. In fact, it declines in volume at about 1 milliliter per year, terrifyingly. What does that mean? Effectively, that means that your brain is shrinking away from your skull. So you would need to turn your stimulator up a little bit for each additional year of age. But there's another way to think about correlation, and that's to take places that we usually use to set our dosage, for example, primary motor cortex, and see if the surface of the cortex correlates in depth with that region of interest. So taking the primary motor cortex, adding a little seed region here at the top, does the depth of the cortex correlate well with that with the depth of that region of interest? The answer is not particularly well. So there are some fairly good R values, this is Pearson's R, of about 0.6 or minus 0.6, so negative correlation is fine too. But they tend to be very closely clustered around the seed region. What about the primary visual cortex? Again, not particularly well. So again, there's clusters of high correlation around that seed region, but away from the seed region, not so good. Okay, so this is a quick tour through some statements about the depth of the cortex across the head. What can we say about this? So one useful sanity check is that the mean depth of the cortex varies in the way we would expect it to. But standard deviation and the coefficient of variation also vary across the cortex. And given that coefficient of variation should be scale-free, it helps us to find areas where depth can be well estimated or poorly estimated. And the answer is actually not very many areas have a good correlation. Oh, sorry, good coefficient of variation. Talking about those correlations then, the depth of the cortex correlates poorly with a seed region in primary motor cortex, so motor evoked potentials, MEPs, are not that good as a predictor of how much energy to deliver to a particular brain region. And that's the same, in fact that's even worse for the primary visual cortex, so setting dosage according to phosphine production is not a reliable way to find the energy suitable for stimulating a nearby brain region. To wrap all this up, so firstly, brain stimulation dosage needs very good prediction of cortical depth. Given that the required dosage changes by this sort of magic value of about 3% per millimetre, we do need to know how deep a particular brain area is. There are other factors, of course, but depth is a clear source of variance. But the way we typically do TMS experiments, which is to find the stimulator output required to generate a motor evoked potential and then set dosage as a percentage of that, actually is not a good way to do it, I think. I think this is what the data here is saying. So motor evoked potentials and visual evoked potentials, or phosphines, are not a good way to set dosage. What that means is that for most applications, we would need a person-specific brain image. So you need to scan your participant and use that in some kind of neuronavigation system to understand exactly how to target a particular brain region with a particular amount of stimulator output. Why are we bothering to do this? Well, so where could we go with this? Well, in the analysis I've shown here, I've only taken people in the sort of middle years of their lives, so 25 to 60 years old, where brain structure is relatively stable. But we can look at people who are older, so if we can see this image properly, so we have people in their middle years have a sort of nicely packed sort of skull, so the brain reaches out to the edges. Whereas in an older person, and by that I mean people in their 90s, the brain is starting to move away from the outer surface, and the ventricles are also increasing, so the amount of fluid in the head is a bit larger. So understanding brain stimulation targeting in aging is a key area, and also in other applications such as eating disorders. So a person with an eating disorder typically has a very different brain structure to others. And of course, another application would be to verify TMS models. So can we use the variance estimate of say, primary visual cortex to map onto the known variance in the stimulator output required to generate visual evoked potentials? So can we map these predictions onto real data? Okay. That was a very quick tour of some depth data. I'd be very happy to chat about this. This is extremely exciting, being able to do this online. So please do join in, or please do get in touch if you're interested. My email and Twitter handle and things are down there. But meanwhile, thank you very much.